Hello students, today we are going to learn about mutation. Now mutation is any change, any alteration in the genetic information. Now this genetic information is present on DNA inside the nucleus. So this genetic information, when it got changed or altered due to many reasons, then there is change in the information. And when there is change in the information, that results in the change in the phenotype and genotype of the organism. So mutation is change in the genetic information leading to what is the genetic material? Genetic material is DNA. So this DNA is present in the nucleus and a single fragment is present on each chromatid, and whenever there is any change in the DNA sequence, the change would result in the change in the RNA, because DNA is changing into RNA, RNA into protein. This is called central dogma. This we are going to learn in our next chapter, that is molecular basis of inheritance. But for to understand the concept of mutation clearly, you should know that this genetic information, whatever is available on DNA, first is getting converted into messenger RNA, and then messenger RNA is utilized, is forming proteins, and these proteins are made up of amino acids. Now these amino acids, they are deciding the phenotypic trait of the organism. So if there is any change in the DNA, that would result in the change in the RNA, that would result in the change in the protein, and ultimately change in the protein changes because of the change in the amino acids. So when there is change in the amino acids, a different kind of protein is formed, and because of this different kind of protein, the phenotypic trait would be different. So mutation is the phenomena which results in the alteration of DNA sequences. Now this alteration results in the, alteration means changes, any change due to any reason, it results in the change in the genetic information. So this alteration results in changes in the genotype. Genotype would be changed. Suppose the genotype is TTRRBB. So due to the mutation, it may change into TTRRBA. So the change in the sequence happens, and because of this change in the sequence, the genotype is changed, and this genotype would result in the change in the phenotype, which we can notice externally. Now, each and every feature in any organism is controlled by one or the other gene. All traits of us, all traits of any organism, they are controlled by the genes. And where are these genes? These genes are located on DNA. And where is DNA? DNA is present on the chromosome. Now, DNA is the carrier of the genetic information from one generation to another. So, genetic material is DNA, which is present in the nucleus on chromosomes, and this is carrying the genetic information for each and every trait which is going to come on in the progeny. Now, any change in this information is due to the change in DNA. If your DNA is changed slightly or completely or to a larger extent, then there will be alteration or change in the genetic material. And this alteration or change in the genetic material is referred to as mutation. Now, these mutations are caused, how, why are they caused? These mutations are caused by mutagens. Mutagens are the chemical or physical factors which can generate mutation. So, these are the chemicals or the physical factors such as ionizing rays or non-ionizing rays, for example, ultraviolet rays. So these are the factors which can generate the mutation. That's why they are called mutagens. Mutagens means mutation generating. they can generate the mutation. They are capable of giving any change, any alteration in the genetic information, which is ultimately going to lead in the change in the phenotype. Now, one DNA helix. DNA is double helix structure. One DNA helix is present in one chromatid. Another is present in another chromatid, and there are two chromatids in one chromosome. This we have learned. Now, one DNA helix runs continuously from one end to the other in each chromatid. And this chromatid is in a highly this DNA is present in the nucleus in a highly supercoiled form. Supercoiled means in coiled structure. This DNA is present in the nucleus. Therefore, loss or gain. Now, suppose any segment of the DNA is lost, or there is any addition of the segment to the original segment of the DNA, this would result in the alteration in chromosome. So chromosome will be changed structurally. The chromosome may become shorter in length, may become longer in length because of the addition or deletion. So there is change in the morphology of the chromosome because of the loss or gain of the segment of DNA, and this results in the alteration in chromosomes. Now, genes are known to be located on chromosomes. Genes are on chromosomes. So, alteration in chromosome results in what? Abnormalities or aberrations, because genes are ultimately responsible for the traits. So, if there is any change in the chromosome, there would be abnormalities or aberrations. Why? Because normal condition is what normal genetic makeup is. And if there is any change in the genetic makeup, that would result in abnormality or aberrations. Now we will see. So this is what mutation is about. Now, what are the different types of mutations? Now we have understood that mutation is any change, any alteration in the genetic information. Now, this genetic information is present in DNA. Now, DNA is on chromosome, and on DNA are genes. So, the mutations can be chromosomal mutations, can be point mutations on the gene itself. So, this types of mutations, first type is chromosomal mutation. That means any change occurring in the chromosome or chromosomes. If any change is occurring in the chromosome or chromosomes, this is the mutation occurs, and this type of mutation is called chromosomal mutation. Now, chromosomal mutation can be further divided into two: morphological and numeric. Morphological mutation is the mutation which is occurring and which is resulting in the change in the morphology of the chromosome. So, morphology of the chromosome is changed in this type of mutation. Another type of mutation in chromosome is numeric mutation, where the number of the chromosome or chromosomes are changed, and because of the change in the normal number, there is mutation. So, first we will do morphological mutation. Morphological mutation are of different types because many things can happen to change the morphology of the chromosome. First is deletion. Deletion means any segment, one segment of the DNA is deleted. So the information would be changed. The genetic information would be changed. Suppose, for example, the original sequence of DNA was A, B, C, D, E, F, and G. And now, because of certain reasons, because of some mutagens, what happened? There is deletion of this segment, C, D, and E. So, what would be the resultant sequence? The resultant sequence would be A, B, F, and G. So, the information is changed. What information was earlier? Now the information is different, and this is mutation. And this mutation has result in because of the change in the morphology of the chromosome, because of deletion of the segment. Now, another thing which can happen is duplication. Now, in this, the segment is duplicated. For example, the original sequence is A, B, C, D, and E. Now, what will happen here? The C, D, E, this segment is duplicated. Now, when this segment is duplicated, the resultant sequence would be A, B, C, D, E, C, D, E. So, again, the information has changed, and this has resulted in the changed information, changed proteins, and changed traits. So, this is because of the change in the morphology of the chromosome because of duplication. Another thing which can happen is inversion. Now, in inversion, what happens? A segment gets cut and inverted. Inverted means ulta. It gets inverted. Now, see this. This is the original sequence. Suppose this is the original sequence. A, B, C, D, E, F, G, and H. Suppose this was the original sequence. Now, after inversion, what happens? This D from here, D, E, F. This is this E, F, and G. E, F, G has been cut and inverted. So what happened? G will come here in the place of E. F is here only, and E will come here. So now the resultant sequence would be A, B, C, D, G, F, E, H. So again the information is changed. So this is because of the inversion. Now, another thing which can happen, which changes the morphology, is translocation. Now, translocation is changing the location. The location of the sequences fragment is changed. Now, here what happens? Two chromosomes are involved, homologous chromosomes. So, what happened? One segment changes its location from one chromosome to another homologous chromosome. As you know that homologous chromosomes are present in pairs. So what happens in this pair? Suppose this is the original pair. So in original pair, suppose one chromosome is having this sequence A, B, C, D, E, F, G, H. And the another chromosome is having this sequence 1, 2, 3, 4, 5, 6, 7, 8. Now, what will happen after translocation? This 6, 7, 8 will change its location from here to the other chromosome. So, what will happen? A, B. Now, what will happen? This 6, 7, 8 will come here. A, B, C, D, E. Now, F, G, H. Between E and F, this 6, 7, 8 will go and get themselves translocated. So this is translocation. The first chromosome is changed, and similarly the second would also change. This is translocation of one segment from one chromosome to another. There is another translocation which is called reciprocal translocation. In reciprocal translocation, both the chromosomes are exchanging their segments. So here there is change in both. There also there is change in both, but here also there is change in both. But here both the segments, one segment is coming from one to another, and similarly the another segment is coming from the second one to the previous one. So this is reciprocal reciprocal translocation. Just see this. The original sequence is A, B, C, D, E, F, G, H. And the original sequence of second chromosome is 1, 2, 3, 4, 5, 6, 7, 8. Now what will happen? 5, 6, 7 of this would get translocated to this one. And from here, E, F, G will get translocated to this. So this is called reciprocal translocation. Both are getting exchanged. So now the resultant sequence would be A, B, C, D, 5, 6, 7, H. And this would be 1, 2, 3, 4, E, F, G, and 8. So changes have occurred because of the reciprocal translocation. So this is how the changes in the morphology of the chromosome occurs. Now just see this diagram. Here the sequence is A, B, C, D, E, F. This is one chromosome. Now what happens? There is deletion of B. So the result would be AC here before this centromere. This is AC and DEF. In duplication, what happens? There is duplication of B. So A, B, B, C, D, E, F. This is duplication. Inversion, it will cut and get inverted. So what happened? A, E, D. A, E, D and C, B, F. So this is because of the inversion. And this is because of the translocation. In translocation, what happened? A, B, C, and in spite of D, E, F, here comes the J, K, L, and D, E, F would come here on the second one. This is reciprocal translocation. So these are the different ways by which the structure, the morphology of the chromosome changes and results in the mutation. Now we will see how the numbers of the chromosomes result in the mutation. Here the change occurs in the number of chromosomes. You know that number is constant for all species, like we are having 46, and these 46 are present in 23 pairs, and these 23 pairs are present in 22 pairs of autosomes and 1 pair of sex chromosome. Now, if there is any change in the number of the chromosome, then it would result in mutation. Now, this is all of two types. First is euploidy. Now, euploidy, the chromosome number changes by complete set. That means the whole set of chromosome number would change. For example, for us, what is N? N is 23. Now, N is haploid. Then it becomes diploid, 2N, and it can become triploid also if the set changes into 3N. So, N, 2N, 3N, and polyploidy. Many sets can be formed. So, when this happens, this type of mutation is called euploidy, where the whole set of chromosome is changed. The whole set is changing. The whole set is doubled or the whole set is single, like that happens in the progeny. And this results in euploidy. This is called euploidy. And this is called another type of numeric chromosome mutation is aneuploidy. In this, what happens? There can be loss of 1 or 2 chromosomes, or there can be gain of 1 or 2 chromosomes. Now, if there is gain of 1 chromosome, what happens normally in a diploid organism? The homologous chromosomes are present in pairs. So, they are present in a set of 2 each. Okay, so if suppose take the example of human beings, we are having 23 sets of chromosomes, and each 23 are present in pair. So number 1 chromosome a pair, number 2 chromosome a pair, number 3 chromosome a pair. Now suppose there is increase in the chromosome of any pair, that results in trisomy. Trisomy is what? It becomes 3. Normally it is pair is 2, but it becomes 3. And this happens due to non-disjunction. What is non-disjunction? If you remember, and in the cell division, meiosis cell division, first meiosis cell division in anaphase, the homologous chromosomes come and settle themselves on the plate. And they move towards the opposite poles, the homologous chromosomes. But in this case, what happens? The homologous chromosomes, they do not get separated, and both will move to one place and in one daughter cell, and the another would have no. So what would be the result? The egg will have two sets, okay, acha, of that chromosome. And now this egg, when it will be fertilized by a sperm, which is having one chromosome of that, so result would be three chromosomes. And this is because of the this process is called non-disjunction. This is failure of separation of homologous chromosomes during meiosis 1. And the classic example of this is Down syndrome. Down syndrome is a disorder in human beings, mental retardation, which is due to the trisomy of 21st chromosome. That means the 21st chromosome, three chromosomes are present. Otherwise, normally should be two, but there are 21st chromosome present, and that results in the Down syndrome. It is a mental retardation in human beings. Now, suppose there is gain of two chromosomes. That means the egg is also having two and the sperm is also having two. So the result would be tetrasomy. That means one set having four chromosomes. Normal is two, but it is having four, and that is called tetrasomy. So trisomy and tetrasomy. Now see what happens when there is deletion of one chromosome. One chromosome is absent. One chromosome is lost. When one chromosome is lost, then what happens? It results in monosomy. That means only one is present. For example, Turner's syndrome. This is also a syndrome in human beings, in human females. So what happens? Loss of one X chromosome. Normally, females are having two X and X. But in Turner's syndrome, there will be only one X chromosome in those human females because of the monosomy. This we are going to study this Turner's syndrome and Down syndrome in detail in genetic disorders. But for the mutation part, you should remember that there can be loss of chromosome or there can be gain of chromosome. If there is gain of one chromosome, it results in trisomy. If there is gain of two chromosomes, it results in tetrasomy. If there is loss of one chromosome, it results in monosomy. And if there is loss of both chromosomes, then it results in nullisomy. So that number would not be there. So from 1 to 23, suppose if 22, both are lost. So 22 chromosome will not be there. So that results in nullisomy. So this is all about mutation due to chromosome, change in the chromosome. There can be change in the morphology of the chromosome. There can be change in the number of the chromosome. Now we will come to the second part, that is change in the genes. Because genes are the codes, the genetic codes which are giving information for the traits. So when there is change in the single base pair, do you remember the structure of the DNA? The nitrogenous bases are present: adenine, guanine, cytosine, and thymine. So if any single base pair is changed, the mutation is resulting due to change in any single base pair, that is gene mutation. Now, gene mutation is also of three types. Point mutation. Now, point mutation is one nitrogen base pair. It can be adenine, it can be thymine, guanine, or cytosine. It gets replaced by another. And before and after codons remain unchanged. For example, the classic example of this point mutation is sickle cell anemia. Now, what happens in sickle cell anemia? In sickle cell anemia, it is caused due to substitution of single base pair at the sixth codon. Now, what is codon? Codon is triplet. Three nitrogenous bases, they form a codon, triplet. We will study about all these things in next chapter. So in this sixth codon, that is coding for beta globin gene, it changes from GAG, that means guanine, adenine, guanine, to GUG in hemoglobin molecule, and that results in the sickle cell anemia. So what is the change? Change is only from this A is getting changed into U. A is changed into U. Change in the single base pair. Now what would be the result? The result is this GAG was coding for glutamic acid amino acid. As I told you that this DNA is changing into RNA, RNA to protein. So every codon is coding for one amino acid. So this GAG is coding for glutamic acid. Now when this GAG got changed into GUG, now it is coding for valine. So the amino acid is changed. When the amino acid is changed, the gene would be changed, and that results in the sickle cell anemia disorder. We will study about this also in detail in genetic disorders. But for this is the classic example of point mutation, where only one base pair is getting changed. In spite of GAG, it becomes GUG. And then the amino acids got changed, and the result, the phenotypic trait would change. The second type is inversion. Now here what happened? It just flipped over. One base get flipped with the other. For example, this GAG changes into GGA. There is inversion of G and A. So G comes here and A goes there. So now this is again different codon, and this codon will code for different amino acid. So there would be difference in the genetic information. Now, the third type of gene mutation is frame shift mutation. Now, what is frame shift? As the name suggests, the whole frame is getting shift because of the change in the base pair. One base pair is changing, adding or deleting. One addition or deletion of base pair changes the complete frame. For example, let's see this. Now, I have taken this sentence. And why I have taken this sentence? Because all words are having three letters. So, three in our codon also, there are three bases. So, this is why did the cat eat the rat. Okay, so three in each. Now this is making some sense. Now what happens? Suppose due to mutation, there is deletion of this D. Now if there is deletion of this D, now what would be the next frame? Why? DIT. Because this this should be IDT, not DIT, IDT. Read it as IDT. This is mistake because this D has been deleted. So now it will become IDT, HEC, ATE, ATT, HER, AT, and so on. So the frame has changed. Now every codon has changed, and every codon will give another amino acid. Similarly, suppose now see there is addition of X. Now this is our original frame, and here there is addition of X here at the place of D, before D. So what would be the result? Y, X, D, I, D, T, H, E, C, A, T, E, A, T, T, H, E, R, A, T, and so on. So this has also changed the frame. So this is called frame shift mutation. The gene mutation is because of the change in the single base pair. It can be point mutation, it can be inversion, or it can be frame shift mutation. So this is all about mutation, and these mutations are altering and changing the genetic information. And so when the genetic information is changed, the proteins are changed, amino acids are changed, and ultimately the phenotype, the phenotypic traits also get changed because of these mutations.